Hello, everyone. So today we're going to talk about glycerophospholipids. So for our real-life biochemistry question, today I brought, have you ever thought that lipids may have more to offer than energy? And I wanted to bring this question because I think that many times when people think about lipid, they think of a bunch of fat. So we already talked about the different functions that lipid have, but I want you to emphasize that lipids are much more than a bunch of fat. So today we're going to see another examples of the importance of lipids. So in our previous classes, we saw that when we have fat acids, we can use them to synthesize triacylglycerol. And this triacylglycerol can be stored at the adipose tissue. So we also talked about the triacylglycerol cycle, when we can have the mobilization of the triacylglycerol from the adipose tissue with the release of glycerol and free fat acids. These free fatty acids, they can go through the blood and get to the liver, and they can be re-esterified to triacylglycerol. But these free fat acids, once they get to the liver, they can also be used for the synthesis of phospholipids. And in this class of lipids that we're going to concentrate today. So it's important to note that the phospholipid, the triacylglycerol pathways, they diverge at phosphatidate. So once we get phosphatidate, we can then synthesize diacylglycerol. And after that, through the reaction of diacylglycerol with acyl-CoA, we can have the synthesis of triacylglycerol. So the liver is the primary site for triacylglycerol synthesis, and then triacylglycerols, they can be transported to be used as energy in different tissues, or to the adipose tissue to be kept as storage. So if you want to synthesize phospholipid, what do we need? So we need the synthesis of a backbone molecule that can be glycerol or sphingosine. We need the attachment of fat acids to this backbone. So here we have the glycerol, here we have the fat acids. Then we need the addition of a hydrophilic head group to the backbone, and the alteration or exchange of the head group throughout the final phospholipids product. So here you can see the structure of a glycerophospholipid. When we have the glycerol, we have the fatty acids, we have the phosphate, and we have the head group. And depending on the head group that we can have different types of glycerophospholipids, okay? So when we have the glycerol as the backbone, then we call this group of lipids, we call them phosphoglycerides or glycerophospholipids. So for example, if I had a head group choline, this is going to lead the production of phosphatidylcholine. So the phospholipid synthesis requires the combination of a diacylglycerol with an alcohol, okay? And then there are two strategies that can happen. One is using an activated diacylglycerol or using an activated alcohol. So let's go for the first step when we use an activated diacylglycerol. So the activation occurs through the reaction with CDP, and then we get the CDP diacylglycerol. Now that we have an activated diacylglycerol, a reaction with an alcohol occurs, in this case is the inositol, and this leads to the production of phosphatidylinositol and CMP. So that's one strategy. The other strategy for the phospholipid synthesis is the combination of diacylglycerol with activated alcohol. So here we have the ethanolamine as an example of alcohol that's going to be activated through the reaction with ATP and CTP, leading to the production of the activated alcohol CDP ethanolamine. So CDP ethanolamine reacts with diacylglycerol, leading to the production of phosphatidylethanolamin. So here just a different representation of what I just said, strategy 1, when we have diacylglycerol activated with CDP, and in this case, the head group activated with CDP, leading to the production of glycerophospholipid. So the most common phospholipid that mammalians have is the phosphatidylcholine. And to produce phosphatidylcholine, we need to get choline from the diet. So choline then is going to be activated through the reaction with ATP, CTP, leading to the production of CDP choline. CDP choline is going to react with 1,2-diacylglycerol, leading to the production of phosphatidylcholine. I just wanted to call your attention that from 1,2-diacylglycerol, through a reaction with acyl-CoA, we can also produce triacylglycerol. So phosphatidylcholine is the most abundant phospholipid. So what if I don't have choline from the diet? Then the organism, there is a way that can be done for the production of phosphatidylcholine. It still happens. So once I have a low port of choline from the diet, phosphatidylethanolamin can be converted into phosphatidylcholine. For this, we need S-adenosylmethionine and the action of the enzyme phosphatidylethanolamine methyltransferase. And this happens in the liver. Once we have phosphatidylethanolamine and phosphatidylcholine, we can have an exchange reaction with serine, removing ethanolamine or choline, and replacing with serine to produce phosphatidylserine. From CDP diacylglycerol, I can also synthesize cardolipin through the reaction with phosphatidylglycerol. So cardolipin, it's a glycerophospholipid that is required for the full activity of cytochrome C oxidase, important for mitochondria function. And it also facilitates the proper folding of alpha-synuclein. So if I don't have the proper folder of the alpha-synuclein, I can have the production of aggregates that are related to Parkinson's disease. So on this side, we can have the CDP diacylglycerol reacting with inositol, leading the production of phosphatidylinositol. And these alcohols can be, alcohol group can be these these hydroxyl groups. They can be phosphorylated, and this can lead to the production of phosphatidylinositol 4,5-biphosphate, which is the precursor of intracellular messengers, diacylglycerol, and inositol 1,4,5-trisphosphate. So how do we decide, how the organism decide if we are going to synthesize triacylglycerol, or if you're going to synthesize phosphatidylethanolamine, or if you're going to synthesize cardolipin? So there is a key enzyme that acts in the regulation of the fate for the lipid metabolism, that is the phosphatidic acid phosphatase, and it's also called PAP. So what happens is that phosphatidate, if you remove the phosphate, we can produce diacylglycerol. And then diacylglycerol can be used to synthesize phosphatidylethanolamine, phosphatidylcholine, phosphatidylserine, or triacylglycerol. On the other side, when we have a lower activity of phosphatidic acid phosphatase, then diacylglycerol is converted to phosphatidate that can be used for the synthesis of cardolipin or phosphatidylinositol. Okay, so you can already realize that if we have loss of function of PAP, this is going to prevent the normal adipose tissue development. And this is going to lead, because then this part of the triacylglycerol synthesis is going to be compromised. And then this can lead to a severe loss of body fat and insulin resistance. On the other side, if I have excess of PAP, then I'm going to produce too much triacylglycerol, and this can result in obesity. So here we have an overview of starting from glycerol or from glycolysis, how do I get to the synthesis of glycerophospholipids? And it's amplified here, the first step, and then the second step that we are going to go through in more detail in our class. So we saw how we synthesize phospholipids, but what if I want to degrade phospholipids? So for that, we need the action of enzymes that are called phospholipase. And these phospholipase, they can act on specific sites. So they can be acyl hydrolases, that is the case for the phospholipase A1, A2, and D, or phosphodiesterase, that's the case for the phospholipase C, and D. So phospholipase 1 and 2, they hydrolyze the ester bond of intact glycerophospholipid at carbon 1 in the case of phospholipase A1 and carbon 2 in the case of phospholipase A2, and both carbon 1 and 2 for phospholipase B. In the case of phospholipase C and phospholipase D, each split one of the phosphodiester bond in the head group, as you can see here. So here, a magnified example of the action of phospholipase C, where we have phosphatidylinositol 5 biphosphate through the action of phospholipase C, leading to the production of diacylglycerol and inositol 1,4,5 trisphosphate. So we already talked about in our other classes, but I want to emphasize here that lipids, they can be used as a form of energy storage. They are part of our membranes, but they are also important for cell homeostasis and signal transduction. So what if I have a dysregulation in lipid metabolism? Well, in this case, I can have metabolic syndrome, of course, but I can also have other disease like Parkinson, Alzheimer, cancer. So this shows the importance of the lipids being much more than just a bunch of fat, as I said at the beginning of the class. What about the lipids and the brain? So when you think about lipids and brain, lipids are involved in neurogenesis, synaptic transmission, signal transduction, neurocommunication. So in nerve cells, lipids represent 50 to 60% of cell membranes, with cholesterol, glycerophospholipids, and sphingolipids being the most represented in the central nervous system. So here you can see the importance of the glycerophospholipids and cholesterol and sphingolipids that you are going to study with Dr. Lewis. So I brought this paper to show the importance of lipids again. And here what they show is when we age, with aging, the level of lipids changes. And in this study, specifically here, but the information they showed on Alzheimer's disease is that if you compare the brain of a patient with Alzheimer's disease with a healthy patient, we are going to see that there are lipid alterations, and these lipid alterations are related to neurodegenerative disease. So for example, we see reduced monounsaturated fatty acids, we see increased saturated fatty acids, increased phosphatidylserine, and decreased phosphatidylcholine, phosphatidylinositol, and phosphatidylethanolamine. And one thing that is really interesting is in this paper they mentioned that once there is these alterations in the lipid profile that you can see in the brain associated with specific disease, we could use this as a biomarker of these lipids as a biomarker for prognosis and diagnosis of different diseases. So this is something that it's really interesting and also shows the importance of lipids. So for today, our learning objectives are to understand glycerophospholipid structure, glycerophospholipids synthesis, lipids function, and lipids change in disease. So I look forward to seeing you in class so we can discuss this further and learn more together.